Hello, everyone. Thank you for joining in tonight on AP Biology Podcast number 2 on local signaling. Tonight we have Lindsay Hara, biology extraordinaire from Harvard University, talking to us about how these types of local signaling work. Lindsay?
Thank you for tuning in, everyone. I'm going to explain to you two different types of local signaling. There is paracrine signaling, and then there is also synaptic signaling. As I refer to my example I have right here, I have my secreting cell in the middle and my different target cells on the outside, and this is all in extracellular fluid. And within these different vesicles within the secreting cell, they're going to form and break free out into the target cell through the extracellular fluid, and my different regulators will go. And this will happen with all my different molecules discharging into the extracellular fluid. So you see, all of them will go to the target cells. Now, an example that we learned in class about this. It's actually very important information. An example in class is like a growth factor. And you see, this secreting cell is allowing these other damaged cells to grow. So this is paracrine local signaling in a nutshell. Now my next example over here, synaptic signaling, is also another type of local signaling. And you see for this one, we have a neuron. This is like a nerve impulse. Our neuron, we have our electrical impulse traveling through the neuron, and then we have the synapse, the distance between the different neurons. It's going to let out a bunch of neurotransmitters out the end of this cell, and it's going to attach into the next cell, and it's going to pass it on, and it's going to go down a long chain as a nerve impulse would. And this is also another type of local. Let me show you again. This is a very important concept. We have our neurotransmitters, really just an electrical signal at this point, going down, and then they change into neurotransmitters, which come off through the synapse. Each one, they're just going to jump up. Now that they have all jumped the synapse, which is a gap in between the cells, they can travel along their merry way down the next neuron. The process will repeat itself. So to keep these straight, paracrine local signaling has to do with a growth, or like in damaged cells. And then also synaptic local revisioning has to do with nerve impulses. Remember, folks, this is a very difficult concept, but it can be made easy through learning. Have a nice night. Thank you for tuning in.